Okay, let's continue talking about the active processes. So there's two types of active processes, active transport and vesicular transport. Both require adenosine triphosphate or ATP to move solutes across a plasma membrane. And that could be for any of the following reasons. The solute is too large for channels, or something's not lipid soluble, or something's not able to move down its concentration gradient, so it needs to move against it. Whereas something moving passively was described as moving with or down its concentration gradient, something that is doing active transport, a solute moving actively, is considered to be moving against its concentration gradient. And this usually requires a carrier protein or a solute pump, something powered by ATP. And there's actually two types of just active transport, primary and secondary active transport. Primary active transport is when the energy required for this transport comes directly from ATP hydrolysis, or the breakdown of ATP. The breakdown of ATP releases energy, and that powers a channel or protein pump in order to allow something to cross a membrane against its concentration gradient. Primary active transport utilizes things like a sodium-potassium pump. This is actually located in all plasma membranes and is involved in primary and secondary active transport. The sodium-potassium pump is the most studied pump. It's called the sodium-potassium ATPase occasionally because it acts like an enzyme and it moves sodium and potassium across the membrane in opposite ways. It actually pumps 3 sodium out of the cell and 2 potassium back into the cell every time it pumps. And we find this very useful, especially in excitable cells like nerve cells and muscles. It really helps maintain electrochemical gradients. So let's look at the functioning of the sodium-potassium ATPase, or the sodium-potassium pump. You can see here it binds to ATP while it's open to the inside of the cell, and it's binding 3 sodium. The breakdown of that bound ATP, resulting in ADP and inorganic phosphate, is going to initiate a change. That change is a shape change. That shape change takes place. Now the protein is open to the outside of the cell and releases its 3 sodium. Then it binds to potassium, and once those 2 potassium bind, it lets go of that inorganic phosphate that it was holding onto, and that initiates another shape change, so now the protein's open to the inside of the cell again. And that's going to allow it to release its 2 potassium and then start everything over again by binding ATP. So let's look at it again, one more time. So we bind 3 sodium, then we use the power of breaking down ATP to cause a shape change. The sodium are released, the phosphate comes off after the potassium bind, shape change, now we release the potassium. So we see a conformation change, which is another name for that shape change, when something happens regarding ATP, either its initial dephosphorylation or breakdown, or the removal of the phosphate from the protein once that's all that is left. And leakage channels are located in nerves and in the cell membranes of muscles. And so we're constantly having sodium go into the cell and potassium out of the cell. So we really do need something like the sodium-potassium pump to work as an antiporter, pumping things across the same carrier in 2 different directions in order to maintain concentration gradients required for the use of muscle and nerve tissues. Secondary active transport is not as direct as primary active transport. It's more of an indirect process where we use chemical gradients created by primary active transport to power secondary active transport. So it really uses a coupled system approach. Doesn't require ATP directly. Instead, we're kind of driving down a molecule using another molecule's concentration gradient. Now, whether it's primary or active transport, it can facilitate either symport movement or antiport movement. Symport movement is transport of 2 different substances in the same direction. Antiport movement is the transport of one substance into the cell while transporting a different substance out of the cell. And we require energy for any type of active transport, whether direct or indirect, but it is more than just the movement of ions. So let's look at secondary active transport. So here, primary active transport has created a high concentration of sodium outside the cell, which naturally wants to leak back into the cell from high to low concentration. If, for example, in this symporter, it also brings glucose with it because both fit, that's utilizing this high to low process or movement of sodium created by primary active transport to bring something else from low to high concentration. So the reason this is active transport is because glucose is moving from low concentration to high concentration, but it's utilizing a high to low movement created by primary active transport. Don't worry about secondary active transport too much. Do know that there's two types and that it's only primary active transport that directly uses ATP. There's also vesicular transport. This is where we transport large particles like macromolecules and fluids across the plasma membrane using cell bubbles or membranous sacs called vesicles. This does require cellular energy or ATP. And there's different ways to do this. There's endocytosis, where we move substances into the cell. Exocytosis, where we move substances out of the cell. Transcytosis, where substances are transported into, across, and then out of the cell. And sometimes just substance vesicular trafficking, where organelles use vesicles to move a substance from one area to another. So here we can see the transcytosis steps, which begin with endocytosis, which is the ingesting of particles into a pit formed at the plasma membrane. That particle will be engulfed by a completed membranous sac, which is now a vesicle. Those vesicles may fuse with a digestive organelle called a lysosome, and what's inside of that vesicle could be decayed and broken down, and then whatever's left might exit via exocytosis or be incorporated into the cell. So it just depends on what the cell is taking in. There's actually 3 different types of endocytosis based on what the cell is taking in. Penocytosis, which is liquid phase endocytosis, where the cell is essentially drinking and taking in liquid-based solutes. Phagocytosis, also known as cell eating, where we see solid particles being taken in by the cell. And receptor-mediated endocytosis, which is really just specific endocytosis. So here's penocytosis, or cell drinking. The cell is taking in solutes dissolved in solvent to form a vesicle surrounding it. Penocytosis is a type of endocytosis, so it is active. It does use ATP, and it's going to take extracellular fluid and solutes into the interior of the cell. A great example of this is how we do nutrient absorption in the small intestine. Phagocytosis is solid phase endocytosis, and we tend to call the vesicle or membranous sac created a phagosome instead of a vesicle, and this is the one that's generally combined with a lysosome. Any of the waste products or residual information or material created can leave or exit the cell via exocytosis. And then we have receptor-mediated endocytosis, where we have these special vesicles that are coated with proteins called clathrin. And this is an endocytosis process usually followed up by transcytosis, but involves the use of special membrane receptors that are only looking for one specific substance. So this might be when the cell needs to uptake a certain enzyme, or bring in a certain lipoprotein, or go after iron, or a hormone like insulin. It's a specific type of endocytosis. So here's a quick review of all 3 types of endocytosis. Phagocytosis is a solid particle being taken in. Penocytosis is a solute dissolved in solvent being taken in. And receptor-mediated endocytosis is when a receptor binds to something specific in a clathrin-coated pit, and that is taken in. But we also can't forget exocytosis. Exocytosis is vesicular transport, so it also uses ATP, but it's a process wherein the cell ejects substances, maybe for hormone secretion, or neurotransmitter release, or mucus secretion, but sometimes it is just the ejection of waste. But here we can see a photomicrograph of a vesicle releasing its contents. And again, this could be waste, it could be a neurotransmitter, but exocytosis is when things exit the cell using vesicular transport. So here is a summary of our active processes. Primary and secondary active transport are going to use as an energy source, either directly or indirectly, as ATP. Exocytosis is going to be when things exit the cell, whereas phagocytosis, pinocytosis, and receptor-mediated endocytosis are when things enter the cell and are all types of endocytosis. All of these, exo, phago, pino, receptor-mediated, those are all types of vesicular transport, so they also use ATP, even though ATP isn't pictured in the kind of images that I've shown for these processes. So next we're going to go back to the plasma membrane and start to look at cell-environment interactions among the plasma membrane now that we know a little bit more about what it can do. So kind of get your plasma membrane hats on so we can talk more about what it looks like, how it interacts with other cells, how it can respond, and the glycocalyx is going to be a huge part of that. It's always involved in this environmental interactions.